So as we've said, digestion of ingested nutrients of lipids requires pancreatic enzymes and bile. Now, the ingested lipids are mostly triglycerides in the form of triacylglycerol or TAGs, and long-chain fatty acids of 12 to 14 carbons. Now, there are 3 lipases which digests lipids. You have acidic lipases, gastric lipases, and pancreatic lipases. Salivary and gastric lipases accounts for 10 to maximum 20% of total triglyceride hydrolysis, while pancreatic lipase is 80 to 90% of total triglyceride hydrolysis. Now, so pancreatic lipase, where is it going to work? It is secreted from the pancreas, but it's going to work in the small intestine. Acidic and gastric is in the stomach, and acidic usually starts in the mouth. So acidic lipases, which can function at low pH, does not require bile acids or colipases, while gastric lipase secreted by chief cells as well does not require bile acids or colipases, and they both yield diglycerides and one fatty acid short medium chain. Pancreatic lipases, however, works at higher pHs because in the small intestine the pH is higher than in the stomach or in the mouth, which is around, in the stomach the pH is around 2 to 3. So pancreatic lipases has a pH optimum of around 7 to 8, and it does require bile salts and colipases for functioning, and it yields monoglycerides and 2 fatty acids. And then the bile salts and the lecithin are used for facilitating long-chain triglyceride hydrolysis and emulsification and solubilization of triglycerides and long-chain fatty acids and monoglycerides. How does emulsification of lipids by bile salts take place? You have a fat molecule, it becomes emulsified fat by what? By bile. Now let's see how a bile salt or a bile acid looks like. It has a hydrophobic side and a hydrophilic side. Now you have a fat molecule over here and you have a bile salt. As we've said, bile salt has a hydrophobic and a hydrophilic part. The hydrophobic part interacts with the surface of the fat molecule, and then the hydrophilic part is the part that interacts with the aqueous environment around the fat molecule, which allows it to be emulsified and be able to solubilize in water environment. You have a triglyceride by pancreatic lipases. It yields 2 monoglycerides and 3 fatty acids, which is what we've said here, monoglyceride and 2 fatty acids. You have the triglyceride pancreatic lipases, which gives you a 1 monoglyceride and 3 fatty acid. But the fatty acid on the monoglyceride is at the 2 carbon. However, if it was a diglyceride and 1 fatty acid, then it would have 2 fatty acids attached to the carbon and then 1 free fatty acid. Now, how to experiment the role of colipases. First, you have a fat droplet and you add to it lipase. The lipase will attach to the fat droplet. But then when you come to emulsify this fat droplet by the addition of bile acids, the bile acid is going to displace the lipase. So the lipase is going to leave and the bile acids is going to take its place all around the fat droplet, which in this way will not be able to hydrolyze this fat droplet. When you added the colipase, they are both attached and interacted with the fat globule. So the colipase added, which binds to both the lipase and the bile acids, allowing both of them to access the lipid-water interface and reducing the pH optimum. Now, how does bile acid form fat fat micelles. So a micelle. You have a fat globule with the addition of bile salt and phospholipid will give you a triglyceride. This triglyceride is going to be emulsified. By the addition of lipase and colipase, it's going to give you a 2-monoglyceride and a fatty acid, which will then form a micelle where the bile salts are all around it, and inside you have the monoglycerides, the phospholipids, and the fatty acids. Now, how does fatty acid absorption in the absence and presence of bile salts? So free fatty acids diffuse down the concentration gradient. They don't need bile salts. Long chain fatty acids are not very soluble aqueous medium, and they need a chyme micelle chyme micelle to solubilize the fatty acid and transport them into the epithelial cell. This is very slow diffusion. And then you have the shorter ones, the short and medium chain fatty acids, which are less than 12 to 14 carbon in size. They are relatively soluble and they don't need a chyme micelle to be able to be transported. So the only one that needs bile salts is the long chain fatty acid. Now, lipid digestion. We've already said that triglyceride, by the help of pancreatic lipases, will give you free fatty acids. Now, you also have cholesterol esters and lecithin. Those are the more hydrophobic. There is no way it will be dissolved alone. The cholesterol ester has cholesterol ester hydrolase, which will then break it down to a 1 free fatty acid and cholesterol. Cholesterol is a more amphipathic lipid. And then you have lecithin, which will be broken down with the help of phospholipase A2 to free fatty acid and lysolecithin, which is more amphipathic. Cholesterol and lysolecithin and other lipophilic substances such as the vitamin A, D, E, and K, which we'll talk about more in the next slide, are incorporated into micelles referred to as mixed micelles. Now, vitamins, which you will take a whole lecture about in biochem, if you haven't already, are divided into two groups. There is the group that is lipid soluble and the group which is water soluble. Now, the lipid soluble ones are not a lot. There are only 4. They are vitamins D, E, A, and K. The way I memorized it is CADE. So CADE, Ka, vitamin K, vitamin A, vitamin D, and vitamin E. Those are the ones that require bile salts incorporated into micelles to be able to be transferred into chylomicrons. While the water-soluble ones are all of the rest of the vitamins, they usually are absorbed by carriers. You have vitamin C, for example, has a transporter called vitamin C sodium cotransporter, or sodium vitamin C cotransporter 1. It takes the ascorbate into the enterocyte using this transporter, and then from the enterocyte into the capillary. Now, vitamin B12 assimilation, it's not that hard if you understand the process and how it moves from one part of the body to the next. It will be easy for you. Just know that it requires 3 proteins: haptocorin, intrinsic factor, and transcobalamin. So first you ingest the B12. It's bound to protein. Okay. The salivary glands, it releases haptocorin. Okay, just know that for now. And then it moves into the stomach. This haptocorin will bind to the B12. Okay. So the B12 bound to protein will move to the stomach. The stomach has acid and pepsin, which will free the B12 from the protein. And then protection from gastric HCl. How do you protect the B12 from the acidic environment? This haptocorin is what's going to protect it. So what's the function of haptocorin bind to B12, it's just to prevent degradation of B12 in the stomach from gastric HCl and pepsin. And then now it moves into the duodenum. The duodenum has a higher pH. Okay? And it also has pancreatic bicarbonate and proteases. This frees the B12 from HC, which is haptocorin. You don't need haptocorin anymore because the small intestine has around a pH of around 7 to 8, while in the stomach it had a pH of 2 to 3. So, one second. In the intestines, the B12 is going to bind to the intrinsic factor. They then form, you need to know as well that the parietal cells release the intrinsic factor. The intrinsic factor is important for B12. You'll always hear cases about how if this patient removed a part of his stomach, he doesn't release intrinsic factor anymore because parietal cells is in the stomach and intrinsic factor is released from the parietal cells. And then he became anemic. And that's why usually people that cut their stomachs, they become anemic later on because they don't have intrinsic factor and they need to take supplements, B12 supplements, which are easier to become absorbed in the human body. Okay? Now the intrinsic factor B12 complex is absorbed in the distal ileum. It's endocytosed, and then the free B12 transported out of the enterocyte. It binds to transcobalamin and it's transported to the liver. Now folate assimilation. You have the pteroyl monoglutamate. It has 3 methods of transportation from the lumen into the enterocyte. You have the first one, which is let's go from here. It's transported into enterocyte by what? The first one is the PCFT transporter, which is the proton-coupled folate transporter. It takes in the pteroyl monoglutamate 1 into the cell by the help of the co-transportation of H+. This is also the H+. How does it keep on moving from inside to outside of the cell to be able to take in more pteroyl monoglutamate is by the sodium hydrogen exchanger. So sodium will go in, taking out the hydrogen, and then the same hydrogen will go in, taking in the pteroyl monoglutamate. And it also has the reduced folate carrier. It'll take in pteroyl monoglutamate and it will take out organic phosphate. Now the pteroyl polyglutamate, how do you hydrolyze it to give me monoglutamate? It's by the folate hydrolase. Okay, this enzyme, it's found on the brush border, and it will give you pteroyl monoglutamate from pteroyl polyglutamate. Now megaloblastic anemia, you'll take it more in depth in HLS in second semester, but from its name, megalo, okay, mega means large, and anemia is fewer RBCs. So megaloblastic anemia is large RBCs and fewer in number. So it will look more like this, when a normal RBC should look like this, and it will be few in number. So you won't have a lot of RBCs. Now vitamin B12 deficiency will cause pernicious anemia, which is the lack of intrinsic factor. It's an autoimmune disorder of the it's an autoimmune disorder where the body does not form intrinsic factor, or by gastrectomy, which is what I've already explained. When you cut the stomach, you cut the parietal cells, so lack of intrinsic factor secretion. Poor development of RBCs in the bone marrow. Patients require supplemental vitamin B12. It can be either intramuscular injection, so an injection into the muscles, or oral vitamin B12 are also effective. Folate deficiency, compromised DNA synthesis, folate needed for DNA synthesis. You should have already you've taken this already. Folate deficiency will lead to neural tube defects. RBCs in bone marrow are also affected. Patient requires dietary changes and supplements. Lack of folate during pregnancy. Major birth defects, neural tube defect affects the brain, spinal cord, and spine. Now, my references for this lecture, it's Dr. Peter's lecture, Guyton Physiology textbook. This is where you'll find most of the diagrams. And Ninja Nerd Science YouTube channel. He has 3 parts for this lecture. It was assimilation of carbohydrates, proteins, and lipids. Thank you for watching, and I hope you understood everything.